Human physiology is built from highly organized sequences of amino acids. When these chains are isolated into specific functional structures, they form a class of medications known as peptides. The clinical baseline for peptide therapy dates back to 1921. Medical researchers achieved a foundational milestone when they isolated insulin, a natural peptide, to regulate human metabolism. For decades since, pharmacology has relied on small molecule drugs. These act as blunt instruments, often inducing systemic effects across multiple tissue types and creating off-target biochemical disruptions. Peptides function on a different mechanical axis. A peptide's physical structure allows it to bind exclusively to a specific receptor site, interacting only with its intended target. Once seated, it triggers a singular signal. Because the interaction is matched, the surrounding cellular architecture remains undisturbed. This specificity is now being mapped to targeted clinical outcomes. Researchers are deploying these molecules to accelerate angiogenesis in damaged tissues, optimize metabolic rate, and repair endocrine signaling. Despite high clinical efficacy, the availability of these treatments is restricted by a complex environment of federal regulations and patent laws. To understand the regulatory and economic forces controlling your access to high-level health and macro-optimization trends, subscribe to the CEO Brief. Deploying targeted signals allows for precise physiological control while avoiding the systemic disruptions common in traditional pharmacology. Advanced interventions are shifting toward triagonists like retatrutide. These molecules are engineered to target 3 distinct metabolic receptors simultaneously. The first mechanism activates the GLP-1 receptor. This slows gastric emptying, which changes how the body absorbs caloric input. The second mechanism relies on GIP receptor activation. Triggering this pathway generates a significant increase in systemic insulin sensitivity. The third mechanism involves the glucagon receptor. As shown on this flowchart, simultaneously activating these 3 nodes forces the clearance of stored lipids in the liver, effectively reversing non-alcoholic steatohepatitis. Reversing this metabolic dysfunction has profound secondary impacts, particularly on the endocrine system. Severe insulin resistance and adiposity can suppress the neurological signals required for spermatogenesis. In many cases, metabolic disease is the direct cause of male infertility. Exogenous hormone replacement, like testosterone, often fails here. Injecting testosterone triggers a negative feedback loop that actively inhibits fertility. Resolving the metabolic block allows the hypothalamic pituitary gonadal axis to autonomously reboot. Once the physiological pressure is removed, natural signal transmission returns. Clinical outcomes show that metabolic optimization can yield a 10-fold increase in sperm count without any direct hormone intervention. Restoring health at the metabolic level allows the body to resume natural endocrine function, addressing the cause of the dysfunction directly. Commercial pharmaceutical deployment relies on FDA-approved autoinjectors. These deliver rigid, standardized dosing formulas designed for the broadest possible population. Clinical precision requires more flexibility. Regulated 503A compounding pharmacies provide modular, individualized dosing regimens outside of commercial patent structures. Compounding allows a physician to break a standardized weekly dose into microdoses. This adjustment can eliminate gastrointestinal distress while maintaining the same total efficacy. For most peptides, oral administration is not a viable option. Gastrointestinal acid rapidly breaks down these fragile amino acid sequences before they can enter the bloodstream. To reach the intended target, these must be administered subcutaneously or into the muscle. Instead, this generated view shows the fluid diffusing directly into the subcutaneous vascular network for deep systemic penetration. Certain repairs require this deep tissue penetration. BPC-157 is often used to trigger localized blood vessel growth, promoting repair in surgical-grade tendon injuries. In contrast, compounds like copper tripeptides function on the surface. GHK-Cu is applied topically to address localized tissue quality. Topical GHK-Cu regenerates collagen and elastin matrices in the skin without ever entering the deeper systemic circulation. Achieving clinical results requires matching the precise biological compound with its optimal delivery vector. The development of these therapies is governed by legal and economic mechanics. The modern framework was shaped by a 2013 Supreme Court ruling. In the Myriad Genetics case, the court established the legal precedent that naturally occurring biological structures cannot be patented. This created an economic hurdle for commercial drug developers. Large pharmaceutical corporations have little financial incentive to fund multi-hundred-million-dollar clinical trials for peptides they cannot own through a patent. Instead, a corporate duopoly dominates the sector, focusing on patented synthetic GLP-1 variants that generate multi-billion-dollar revenue streams. Federal oversight of compounding tightened following a 2012 contamination scandal in New England. The subsequent investigation triggered strict new regulations on custom-made medications. Starting in 2014, the FDA allowed a specific list of Category 1 peptides to be produced by compounding pharmacies under strict quality standards. That classification was reversed in 2023. The FDA recategorized 19 popular peptides as Category 2, effectively ensuring they were banned overnight. This regulatory shift followed lobbying efforts by patent holders to limit competition from unpatentable compounded alternatives. The legal status remains in flux. Upcoming committee reviews are currently assessing whether 7 of these peptides will be re-legalized. Ultimately, the availability of these treatments is dictated by legal maneuvering and the economics of patent protection, rather than clinical evidence alone. The current regulatory friction has led to an explosion of misinformation within the fitness and biohacking communities. There is a common misconception that molecules like IGF-1/LR3 will generate significant muscle mass on their own. Without resistance training, these peptides provide no meaningful growth. More importantly, the 2023 ban has driven many users toward a dangerous and unregulated gray market. Purchasing compounds labeled research use only carries extreme physiological risks. These vials lack standardized quality control. Users risk exposure to incorrect molecular sequencing or lethal endotoxin contamination. Ensuring safety requires sourcing these molecules exclusively through regulated 503A compounding pharmacies. Deploying these tools effectively requires the active management of a specialized medical professional to monitor dosage and physiological response. As medical technology advances, high-level health optimization is increasingly defined by this level of molecular precision. Subscribe and turn on notifications to stay informed on the research, and leave your perspective on the current regulatory environment in the comments below. While the biochemical ability to address physiological dysfunction exists, reaching its potential requires navigating a complex environment of regulatory and corporate friction.